Hello, could somebody confirm you can hear me?
Yes, I can hear you.
Right. Are there any questions before we get started? I guess I can leave that here. All right, if there's no questions, let's begin. Today is August 17th, and we should be on chapter 8. We should finish chapter 8 and start chapter 13. I'm not sure where we're doing on that. Please remember your infectious disease paper is due this Saturday at 11:59 p.m. Any questions about anything? Today's Thursday, so I don't need to tell you about the quiz. Our last quiz will be next week.
And is it actually only on chapter 8 and lab 9?
Yes,
Okay.
quiz 5 is the most narrow focus quiz. Of course, we're spending more time on chapter 8 than we are in most of the other chapters. All right, let's go to the lesson. I have that we were talking about negative selection. Yeah, we haven't talked about recombination yet. Just a minute here. I've got a frog in my throat. So last time we talked about the different ways you could test for a mutant positive selection and negative selection. And with negative selection, what you're doing is you're replica plating the colonies using a velvet stamp, and then you're stamping the colonies onto two different media. And if a colony doesn't grow on one media but does grow on the other, we can say that colony is a mutant colony. Any questions about any of that? Okay, let's move on to recombination. Genetic recombination is the exchange of genes between two DNA molecules. It is similar to, but a little different than crossing over. Crossing over is when two chromosomes get together side by side, and almost always the chromosomes which cross over are going to be the same pair, like chromosome 1 from the mother and chromosome 1 from the father. And then the chromosomes literally cross over. And you'll note that what did I say? The mother was blue. The mother is lowercase and blue. The father is uppercase, jeans and white. The chromosomes cross over and then they exchange parts. So it breaks right here, and then this part of the father's chromosome joins to the mother's, and this part of the mother's joins to the father. So that, let's see, this is the father's chromosome. Well, it's white, so that's correct. But the lowercase, they for some reason switched the letters there. But that's the father's chromosome, and you'll notice that right here, it switches, and now it's the mother's chromosome. And the same there. There's the mother's chromosome, and it's got a piece of the father's chromosome. Crossing over is when two DNA molecules pair up and then cross over and literally break and exchange pieces. It is similar to recombination, but it is a little different. And that is all cases of crossing over involve recombination, where the capital H gene and the capital I gene, for example, recombine, and now they're with the lowercase g gene instead of the uppercase G gene. Any question about that? So crossing over always involves recombination, but recombination is a little more general, and it can be the exchange of genes between any two DNA molecules. It doesn't have to be an equal exchange. For example, if a piece of a chromosome were to break off, let's say chromosome 1, and then combine with chromosome X, that would be recombination. It is not crossing over. Any question about any of that? The point of recombination is it makes new gene pairs. Previously, the uppercase H gene was only aligned or with on the same chromosome as the capital G gene. And after recombination, we now have the capital H gene with the lowercase g gene. So that makes new genetic combinations, shuffling the genes within an individual. Any question about any of that? Why this is important to populations is it allows new genetic combinations to be made, which may give the individual a selective advantage. For example, early on in human evolution, the blonde hair allele was with the blue-eyed allele. And they always were on the same chromosome, so they always went together. Those two genes are now unlinked, and you can have the blue-eyed gene with the dark-haired gene, or brownish hair or black hair gene. And it just allows new combinations within the population. So it shuffles the genes within the population. Any question about any of that? Let's talk about genetic transfer. Vertical gene transfer occurs during reproduction. So between generations of cells, the parent cell will give its genes to its daughter cells. When we're talking about reproduction of humans, two parents will give their genes to one offspring. And that's vertical gene transfer. Horizontal gene transfer is the transfer of genes between cells of the same generation, where a donor cell gives its genes to a recipient cell. Horizontal gene transfer can happen in three different ways: by transformation, conjugation, and transduction. And I should mention these three ways happen in prokaryote cells. Like in humans, we don't really have sexual conjugation. I guess we do have transduction. It just happens very seldom in the germline. And then transformation doesn't really happen in human cells. But in prokaryotes, we have all three ways going on. Transformation is where DNA comes out of one cell. Usually, that cell dies. The DNA is then released into the environment and is naked DNA. The DNA can be taken up by a recipient cell. And then the recipient cell, let me blow this up, can express some of that DNA that it took up. What happens is that the DNA taken up by this cell has to recombine with the chromosome. So the lowercase a gene has to be inserted into the chromosome. Usually that happens by crossing over in genetic recombination, so that if the lowercase a gene is taken up by the chromosome, then the uppercase a gene is lost. But anyways, that's transformation, where DNA is taken up by a recipient cell, but DNA usually comes out of another cell, and that other cell usually dies. That doesn't have to be the case, but that's usually the way it goes. And then the DNA is taken up into the chromosome, and then it's expressed. Usually, not all of the DNA that's taken up is expressed. For example, in this case here, the lowercase a, b, c, and d genes are taken up by that cell. Only the A gene is incorporated into the chromosome and then will be expressed. The importance of this is, let's say the lower A gene is a gene for drug resistance. Let's say it's just penicillin G resistance. This cell now will become penicillin resistant when it takes up the lowercase a gene and then expresses it. Any question about any of that? Here we're looking at transformation. The cell incorporates a piece of DNA, takes it in. That DNA, some of it is expressed, and that changes the phenotype of this cell. Transformation is the process of taking in DNA, but we can also say that this cell becomes transformed because its phenotype changes. So transformation is used in both ways. I will use it to mean the process of taking in DNA and then expressing that DNA. Okay, any question about any of that? All right. The second way that cells can have horizontal gene transfer, where DNA moves from one cell of the same generation to another cell of the same generation, is by conjugation. Conjugation happens in two ways, depending if it's a gram-negative cell or a gram-positive cell. In gram-negative cells, conjugation happens by this cell here. Let me blow this up. Makes a sex pilus, and then the sex pilus literally skewers that cell and acts as a winch to bring the two cells closer together. And then DNA from this cell moves into that cell. Any question about any of that? We'll talk a little bit more about this one here. That's what happens in gram-negative cells. In gram-positive cells, sexual conjugation can happen by a mating bridge. What happens is two cells come together, and then they lose their cell wall in this location, and then the cell membrane fuses. So instead of the cell membrane coming across this way, the cell membrane goes across this way and then fuses to that one. So the cell membrane goes that way, and then this way and that way, meaning there's a connection between the cytoplasm, between this cell and that cell, so that plasmids can move, or DNA can move from one cell into another cell. So let's say it moves from this cell into that cell, but in reality, the DNA could move the other way. That's what a mating bridge is, which happens in gram-positive cells. Any question about any of that? In both ways, DNA can move from one cell to another. Obviously, the two cytoplasm are connected here, so DNA can move from one cell to another, especially if it's a plasmid DNA. Here, the plasmid DNA moves from this cell and can actually go through the sex pilus. The sex pilus is hollow, and the plasmid can move through it. Any question about any of that? Let's talk about sexual conjugation in a little bit more detail with the gram-negative cells. As I stated, the sex pilus from this cell, the F+ cell, makes the sex pilus, which then skewers this cell. And usually they're not this close when they skewer, they're far away like this. And then the sex pilus acts like a winch and can bring the two cells closer together. They do not have to be this close together. The sex pilus is hollow, and the DNA plasmid from this cell can move into that cell through the sex pilus. What happens when the two cells are connected by the sex pilus is this cell has a plasmid called the F plasmid, which has the F genes on it. Not terribly creative in the words here, but one of the genes on this plasmid codes for the production of the sex pilus. And then this plasmid replicates independently of the chromosome. And then one copy of the plasmid moves into the other cell. This cell we call F minus because it doesn't have the F plasmid, doesn't have the sex pilus, which is coded for by the F plasmid. So there we see the plasmid replicating and moving into this cell. That changes the phenotype of this cell. This cell now has the F plus plasmid in it, and it will grow a sex pilus. And then this cell we call an F plus cell can go out and skewer the F minus cells, and then the process can repeat where the F plasmid can replicate and then move into the F minus cell. Any questions about any of that? Obviously, I'm glad nobody asked me why, because it occurred to me, I don't know why. It's just the way the F plasmid works. It makes the sex pilus, which allows the plasmid to move from this cell into that cell. And that transforms the phenotype of this cell. So it's no longer F minus, it's now F plus. Any question about any of that? If you're wondering why this is called sexual conjugation, all you need to do is ask your significant other. Any questions about that? Humans don't really have transformation because DNA, if it were to leave another human cell, it's not going to get through the skin or even through the mucous membrane. So it won't be incorporated into most other cells. We also don't have sexual conjugation, although our reproduction is sexual reproduction, which is a little similar. The difference is it's not a plasmid moving from one cell to another, and it's not DNA. Well, it's not really DNA moving from one cell to another. It's two cells, two gametes fusing with each other. So we don't really have sexual conjugation. The third way that genes can engage in horizontal gene transfer is with transduction. Transduction uses a virus, and we haven't talked about viruses yet. So we need to introduce viruses. Transduction can occur in human cells. However, it's very unlikely that transduction would ever occur from one cell and then move into a gamete of an individual. It may happen in the nose cells, for example, where a human nose cell picks up DNA from transduction, but who cares? That's not going to be passed on to your offspring, and only one cell of your nose will be expressing a gene from another cell by transduction. So like I said, it probably happens in humans, but who cares? Because most of the time it probably happens in the nose cells or in the throat cells. And it very, very rarely would ever happen in a gamete. And unless it happens in the gamete, It's not important to the population, meaning it doesn't change the genetic makeup of the species. Any question about any of that? Well, transduction does happen in bacteria. Let me blow this up. What happens is a cell becomes infected with a virus. We're talking about generalized transduction. The virus, when it infects the cell, will then inject its nucleic acid into the cell. And we're going to talk about it as a DNA virus just to make it a little easier. This DNA virus will then want to run what's going on in the cell and make copies of its genome and make copies of the virus. And the easiest way for the virus to gain control of this cell that it's infected is to degrade the host chromosome. So early on in the infection of this cell, one of the first genes expressed is a nuclease, which goes in and cuts up the host cell's chromosome. Now, the genome which is running the shots in this cell is the viral genome. And the viral genome will make copies of its genome. And this is DNA molecule, so we'll say it's replicating its DNA molecule. And then that DNA is being transcribed and translated to make the viral proteins. And then the viral proteins get together with the viral DNA to make the new viruses. And then the last thing this virus does is it codes for the destruction of this cell. And when this cell bursts, it releases the newly replicated viruses. And then they will go on and infect other cells. And that's just the life cycle of a virus. We're talking about one type of bacterial virus, but you're getting the picture because it's mostly this virus which does engage in transduction. Well, in viral replication, mistakes get made, and viruses aren't terribly worried about mistakes because, well, viruses make a lot of offspring. And if one of them is a mistake, the virus isn't worried because it's got lots of offspring. Let me see if I can blow this up. You'll note that the DNA pieces of the degraded chromosome are dark purple, and then the viral DNA copies of the genome are reddish purple. Well, look what DNA got inserted into this virus and into that one too. That is not the viral DNA, which is a reddish purple, which is shown in all of these viruses. It is the purple of the host piece of DNA. And now what happens when this virus infects this cell? It goes through the normal viral mechanism, injecting its DNA into the cell, but this is not viral DNA. It is DNA of this host cell. And then that DNA can recombine with the host chromosome. And when it's on the host chromosome, this DNA from this cell can now be expressed, changing the phenotype of this cell. So this is a recombinant cell expressing the DNA of this cell. Any question about any of that? So transduction happens with a virus, and then a mistake gets made. Instead of the viral DNA being incorporated into all the viruses, one or a few get the host chromosome DNA, and then that's injected into this cell, and then this cell picks up new genetic information. This is important for the species because let's say that the DNA being incorporated into this cell gives the cell antibiotic resistance, something like penicillin G resistant. This cell, which was now sensitive or was sensitive to penicillin G, is now resistant to penicillin G. Any question about any of that? All right. Well, let's talk about how often these events occur. You could, for example, have the development of mutation give rise to antibiotic resistance. Let me see if I can do that. AB for antibiotic. I didn't get that. I think I can do this. So mutation gives rise to antibiotic resistance. How often does this happen? Well, we know this regular spontaneous mutation rate is this will happen about one time in every 10 to the 6 genes. Oops, wrong way. And that's just the rate of spontaneous mutation. When we look at how often this occurs with transformation can spread around antibiotic resistance about 1 times 10 to the 5th, in this case, cells. I need to make some of these. Come on. There we go. And how often does sexual conjugation happen in bacteria? We're talking about gram-negative bacteria because I'm not sure about how often the mating bridge occurs. But sexual conjugation also spreads around new genes like antibiotic resistance about one time in every 10 to the 5th cells. And then transduction. Spreads around something like antibiotic resistance about the same times, about 1 and 1 to the 10 to the 5th cells. So transformation, conjugation, and transduction are very important to the population of bacteria, because if they want to develop antibiotic resistance, like let's say it's a bacteria in a hospital where there's lots of antibiotics, it's most likely going to get it by transformation, conjugation, or transduction, because it's going to happen 10 times more frequently than it will happen by mutation. Any question about any of that? Now, with that stated, if there is no genetic variation within the population, then new variants have to arise by mutation. So the very first time a gene can appear in the population, it has to arise by mutation. And we actually have an example of that I've already given you. Over 10,000 years ago, there were only brownish-eyed individuals in the human population. all of the genes were brownish. So people either had very dark black eyes or brownish eyes or even hazel brown eyes. Okay, but they were all brownish. And then 10,000 years ago, we had a spontaneous mutation giving rise to the blue-eyed allele in the human population. And that's the only way that new genetic variation can occur in a population. But once that variation exists, in prokaryotes, we're talking about, this variation is most likely to spread by one of these three mechanisms, either by transformation, conjugation. I got that. For some reason, that's small case, but that's all right. Let's go here and do 3 and bring that up. There we go. So once the variation exists, it's more likely to spread by transformation, conjugation, or transduction, and will happen 10 times more frequently than it will happen by mutation. Any question about any of that?
So then is it no longer a mutation?
Well, it depends what you call a mutation. If you want to call an individual with blue eyes a mutant, that's not incorrect because 10,000 years ago, it was a mutation. But today, it's the part of the human normal population and it's normal variation within the human population. And like I said, it just depends what you're talking about and what you mean. Am I clear on that?
So there's never a point in time when after a certain amount of time goes by that it's continued having that same mutation that it's considered normal?
No, I'm not saying that. Every once in a while, I have no idea how often, but I'll bet every once in a while, a brownish eyed individual will have a mutation and then they will produce the blue eyed allele because that's going to happen every one time in 10 to the 6 individuals. And the human population is over 1 million, right? In fact, it's almost 8 billion. So in the eye color gene, every once in a while, the brownish-eyed allele will mutate to the blue-eyed allele, and vice versa. The blue-eyed allele will mutate to the brown-eyed allele. It's not going to happen very often. Like I said, it's only going to happen maybe in 1 in 1 million children. And for a brownish-eyed parent, they'd probably never notice it because they'll just say, oh, I guess we both had the blue-eyed allele because we had a blue-eyed child baby. But if it ever were to happen with 2 blue-eyed parents and then they had a brown-eyed parent, they could say, well, I guess that was mutation. The trouble is you'd have to prove the paternity of the child. But if the paternity was the father and both the father and the mother had blue-eyed, that would be proof of a mutation. Okay, so it does happen. Spontaneous mutation does happen. It just doesn't happen very often. Does that answer your question?
Sure.
Well, hopefully I answered the question. All right. Anyways, that's the way that genes can be spread around in the population. Plasmids are important because a plasmid is much more likely to move from one cell to another cell than the DNA and the chromosome. Plasmids, we've already talked about them a little bit, but they're a small circular piece of DNA which is outside the chromosome, what we mean by extrachromosomal. What's different with the plasmid compared to the chromosome is the chromosome has thousands or maybe even tens or hundreds of thousands of genes on it. And a plasmid only has a few genes on it. So let me blow this up and let me see if I can count the number of genes on here. There's 1, 2, I don't know what that stands for. That might be just cut sites. 3, 4, maybe 5 or 6 genes on this plasmid. And that's all there is. On the chromosome, there's many, many more genes, and it's a much longer stretch of DNA. Plasmids do replicate independently of the cell's chromosome. We actually saw that here, and I did mention it right there. This plasmid is replicating independently of the chromosome, making a copy of itself, and then one copy moves into the F- cell, changing it to an F+ cell. So that's the importance of plasmids. They are more likely to move from one cell to another than the DNA on a chromosome. However, plasmids can combine with the chromosome and then be expelled from the chromosome, allowing the plasmid DNA to switch. And so this is just a way of shuffling up the DNA in a cell. And that is, if the cell has a plasmid, it'll have certain genes on it. And then some of those genes can be put on the chromosome, and some of the genes on the chromosome can then move to the plasmid. Any question about any of that? All right. This slide is showing you that there are different names for different plasmids and they serve different functions. All you need to know is there's different types of plasmids. You do not need to know the names of the categories of the plasmids. A transposon is sort of like a plasmid in a sense, but it is actually a jumping gene, a piece of DNA that moves around in a cell from one location to another. They were first actually discovered in corn, where there was a gene in the corn chromosome, or one of the corn chromosomes, and they noticed that gene was changing its location, even changing its chromosome number. And they called that a transposon, a jumping gene, a segment of DNA that moves from one region of DNA to another. Now in bacteria, a bacteria usually only have one chromosome. So usually a transposon moves from one location in the chromosome to another location. But it can move from the chromosome to a plasmid, or vice versa, from a plasmid to the chromosome. And then in humans, or any other eukaryote, the transposon can move from one chromosome to another, or it can move from one location on one chromosome, let's say the X chromosome, to another location on the X chromosome. So that's what a transposon is, a jumping piece of DNA that moves about. A transposon always includes the transposase gene and then inverted repeats. The transposase gene actually helps the cell cut out the DNA so the transposon can be excised from this region of the DNA, and it also helps anneal the DNA back together. And then it comes to another location and cuts the DNA so the transposon can move in that new location. The inverted repeats help the DNA to insert themselves in a new location. Any question about any of that? So transposons always have the transposase gene, and they have inverted repeats on both sides of the transposase gene. And that's the minimum that a transposon can be. There are more complex transposons that carry other DNA on the transposon. Like TN5 is transposon number 5, which carries the kanamycin resistance gene. Usually, if there is another piece of DNA on a transposon, you'll note that the other DNA is in between two sections of the transposon. So this would be the normal section of the transposon, including the transposase and then the inverted repeats. We have a transposase gene here, the inverted repeats there, and then a transposase there, and a inverted repeat there. Okay. For a transposon, you don't need two transposase genes, but this is just the way transposons work. But you do need an inverted repeat over here and an inverted repeat over there. You always have to have that. You always have to have at least one transposase. And usually there's a complete set of transposon over here and one over here. And then this works as one transposon. Any question about any of that? All right. Well, transposons are really important for cells because they're shuffling DNA around in the cell. They can move it around in one location to another, one chromosome or part of the chromosome to another, and they can also move this DNA from the chromosome into the plasmid or vice versa. And I stated that with plasmids, why they're important is a plasmid is more likely to move from one cell to another cell than the chromosomal DNA. And so transposons, if they move to a plasmid, here's one that did move to a plasmid, then this plasmid is more likely to move into another cell. And this plasmid is carrying the kanamycin resistance gene in this transposon. So plasmids and transposons help shuffle the DNA in a cell, and then the plasmid helps the cell exchange that DNA from the cell it's in to another cell. Any question about any of that? All right. So genetic variation is important to populations because if you didn't have genetic variation and then the environment changed, the species would be in trouble. Actually, this is true for polar bears. They are well adapted to living in the Arctic. However, the Arctic is now warming, and the behaviors and adaptations that the polar bear had for the Arctic need to switch. But the polar bears are low in numbers, and so they don't have much genetic variation. And it's questionable whether the species can survive because they don't have the genetic variation to survive a changing environment, one that is getting much warmer. For example, polar bears had a very good technique for getting food. They would find a seal hole, which is a hole in the ice that the seal makes, and it comes up to breathe. And the polar bear would just find the seal hole and then watch for the seal to come up and breathe. And then the polar bear would get an easy lunch, meaning would try and trap or catch the seal when it was trying to breathe. And obviously the seal has to come up to breathe, so it has to use the seal hole. Well, now with the Arctic melting, the seals don't have to even make seal holes, or they certainly don't have to use it. They can come up in many different places to breathe. And so the polar bear's solution for getting an easy lunch no longer works. And whether the polar bear will survive or not remains to be seen. It depends on how quickly the polar bears can get genetic variation, how much genetic variation they have, and how quickly the Arctic sea ice melts. So genetic variation is important for populations, and genetic variation, if it does not exist in the species, it can only arise from mutation. But once the genetic variation arises in the species, it can be moving around by crossing over, by the plasmids moving from cell to cell, or plasmids moving it from one location in the cell to another, and the same with transposons. And so crossing over doesn't create new variation in a cell, but it's important for making new genetic combinations. And then the variation, if it exists in the population, it can spread by either transformation, conjugation, transduction, and then transposition increases that spread. All of this genetic variation drives evolution, which happens from natural selection. Like in the Arctic, the natural selection that's going on is there's selection for certainly polar bears that can get a lunch more easily than just waiting for the seal to come to the seal hole. They now have to catch their lunch. And that's evolution of the polar bears in the Arctic. Any question about any of that? All right, that's it for chapter 8. If there's no questions, let's move on to the next chapter. Let me make sure I save this. Our next chapter is chapter 13, which I think are viruses, viroids, and prions. So chapter 13, viruses, viroids, and prions. This slide is showing you the major goals and a rough outline. Know the terms. What's a capsid? What's an envelope? What is a spike? It's a viral spike. What is a bacteriophage? What is the lytic life cycle? What's the lysogenic life cycle? What's a latent life cycle? What's a persistent viral infection? What is a prophage? What is a provirus? What are prions? And what are viroids? I changed the order there just to make the life cycles together. Understand that the characteristics of viruses, what their structure is, and then the classification of viruses. Understand about viral multiplication. And lastly, understand what prions are, understand about them, and then understand what viroids are. Any question about any of that? All right. The only goal a virus has is to replicate. How do viruses do that? They have a general strategy where they invade their host cell and then take over that cell, using that cell to make copies of the virus. When we say they take over the cell and use it to make the copies, we say that the virus is hijacking the cell. And then the cell is forcing it to make viral copies. The viral copies then escape from the cell. Let's watch a little video of this where the virus will be taking over the host cell. Phage is a short term for bacteriophage. We'll talk about that later.
In the microscopic world of bacteria, a miniature act of piracy is about to take place. T4 bacteriophages. With their spider-like tail fibers and prism-shaped heads, these viral marauders plan to hijack an unsuspecting bacterium and use it as their own metabolic nursery. This E. coli bacterium has seen its last day. Binding to its surface, the virus injects its precious cargo of genetic material into the cell, commanding it to produce a new batch of T4 bacteriophages. In a final act, the bacterium is forced to self-destruct, releasing hundreds of viruses ready to kill again.
Any questions about that? I think we'll leave this up because I'll probably show you another video later on. Oh, well, maybe not today. So some general characteristics of viruses. Viruses are inert when they're not within their host cell. In fact, they display little or no metabolic activity outside of their host cell. They only have metabolism when they are inside their host cell. The host cell is a cell that they can infect and then replicate in. Viruses have to live within a host cell. So we can say they are an obligate intracellular parasite. They cannot reproduce, and in fact, they cannot have metabolism going on unless they're inside their host cell. They multiply within their host cell, and they use the host cell's machinery and molecules to replicate, meaning they can use the host cell's enzymes, nucleic acid, amino acids, etc. Viruses have no ATP generating mechanism. They rely on their host cell to supply the ATP the virus needs. Viruses do have a genome, and that genome is either DNA or RNA. It is never both. This is unusual for cells because cells only have DNA for their genome. But some viruses have RNA for their genome. The genome is the centralmost molecule of a virus. And here we see a virus with this part of the virus cut out so you can look inside. And we see the viral genome inside. Surrounding the viral genome is a protein coat. Protein coat always surrounds the viral genome. The protein coat is made up of capsomeres, and we call the protein coat a capsid. This virus, let me blow that up a little more. Maybe I can do it from here. This virus is composed of one capsomere right there, which repeats there, and all of these. So this virus is made up of one, or the protein code of this virus is made up of one protein. That isn't always the case. There are several viruses which have several different proteins in their protein code. But this one, it's only showing one protein in the protein code. Some viruses, besides having a protein coat surrounding their genome, they also have a lipid envelope which surrounds the protein coat. So if this virus were to have a lipid envelope, it would be in this region here, surrounding the protein coat. Not all viruses have a lipid envelope. Only some viruses have a lipid envelope. That lipid envelope is a lipid bilayer, and we'll talk more about that later. Any question about any of that? All right, some general characteristics of viruses. All viruses have a host range. This is the range of cells that the virus can infect and replicate in. The host range is determined by specific host attachment sites that we call receptors. So this cell is the host cell for this virus, and it'll have a receptor on it that the virus binds to. There can be a molecule in the cell wall. If the cell doesn't have a cell wall, it would be a molecule in the cell membrane. If the virus were to bind to the flagella, it could be a molecule in the flagella. Whatever the virus binds to, we call it a receptor. The molecule of the virus that binds to the host cell receptor is called either a ligand or an adhesion. Both terms are used to describe the molecule of the virus that binds to the receptor. And that makes the host cell binding very specific. You have to have certain molecules on the virus that bind to certain molecules on the host cell. If the host cell doesn't have that receptor, the virus will not bind to that host cell. And if it doesn't bind to the host cell, it cannot infect the host cell. Any questions about any of that? Most viruses only infect one or a few cells of one or a few species. Like HIV does not infect all human cells. It does infect T lymphocyte. It can infect the brain. It can infect, I used to know a third one, macrophages, I think is what it infects. It can infect those cells, T helper cells. It does not infect T killer cells. It does not infect a B lymphocyte. It does not infect, as far as I know, muscle cells. The point is HIV only infects certain cells of humans, and then there's one other species that HIV can infect. It can infect one of the chimpanzee species. There's two species of chimpanzee, and I don't know which chimpanzee it can infect, but it can infect one of the chimpanzees. Most viruses are that specific, where they only infect one or a few types of cells of one or a few species. There are some viruses which are not so specific, like bird flu, for example. I'm trying to think of another one. Encephalitis virus would be a good example. What do you call it? Eastern equine encephalitis would be an example. This virus can infect human cells. It certainly can infect human nerve cells because it can cause encephalitis, so that's a brain cell. I'm not sure what other cells it can infect in the human, but the point is it can infect human cells. It can infect most other mammals, like horses and pigs, and it can infect small rodents. That's actually where the virus tends to live, in small rodents like mice and rats. And then it can infect mosquitoes, and then the mosquito brings it to us. Any question about any of that? Oh, I think it can infect birds too. So this just tells you it's infecting a large number of different animals. Most mammals, certainly humans, small rodents, birds, and at least mosquitoes. But most viruses are more specific in that, and they only infect cells of one or a few species. Like HIV only infects 2 species. As far as I know, the human colds only infect 1 species, although there could be some that infect other animals. Polio virus only infects humans. Any question about any of that? How are we doing time-wise? Oh, we got a long time to go still. Here we're looking at the size of a virus compared to the size of a prokaryote. E. coli is a little bit larger than most prokaryotes, and then there's a prokaryote next to a red blood cell. As you can see, viruses are much smaller than E. coli, and E. coli is much smaller than a red blood cell. The smallest virus that you guys have ever heard of probably is poliovirus. There are a few viruses that are a little smaller than poliovirus. Where do I have poliovirus? Around 30 nanometers. We can get down to a virus which is as small as 20 nanometers. But most of you have never heard of bacteriophage MS2. Actually, it's 24 nanometers, not 20 nanometers. The largest virus is megavirus, and it's approaching the size of the smallest prokaryote cells. So the largest viruses are around 0.4 micrometers in size, so 0.4 micrometers, and that's about the size of the smallest prokaryote cell. Ebola virus is a little smaller than megavirus in its diameter, but in its length, it is the largest virus, where it is about 1 micrometer long, just a little shorter than 1 micrometer long. But in its length or width, it's very narrow, and so its diameter is smaller than megavirus. Here we go. The sizes of viruses range from about 20 nanometers to 440 nanometers. In micrometers, that diameter would be 0.02 micrometers to 0.44 micrometers. And in their length, they can be longer than that. The largest being Ebola virus, which can be about 1 micrometer long. But like I said, in its diameter, Ebola is not the largest virus. Megavirus is. When we're talking about viruses, it's important to talk about viral structure. The structure are the different parts of the virus. A virion is a single mature, complete infectious virus particle. So normally when we're talking about a virus, we are talking about a virion. Viruses contain a nucleic acid for their genome. However, viruses are unusual in that the DNA can be their genome or RNA can be their genome. Their genome is never both DNA and RNA. It's one or the other. The DNA can be double-stranded DNA, and that's normal. Most of the time, our cells have normally have double-stranded DNA, but the viral genome could be single-stranded DNA. That is also normal in our cells. Whenever the DNA is being replicated or being transcribed, the DNA is single-stranded. The viral genome of viruses can be RNA, and there can be double-stranded RNA genomes for the virus. This is unheard of in our cells. Our cells have RNA, but it's never double-stranded RNA. The viral genome can also be single-stranded RNA. If we have a viral genome which is single-stranded RNA, it's important to state whether the strand is the positive single-strand RNA or if it's the negative single-strand RNA. The positive single-strand RNA viruses can have their genome be read by a ribosome. The negative single-strand RNA genome viruses cannot have their genome read by the ribosome. And that's because the ribosome can only read RNA if it's positive strand. So positive strand the RNA will eventually have AUG in it, and then the ribosome will begin protein translation at the AUG site. The negative strand, single strand RNA viral genome will not have AUG in it. It will instead have the corollary, which will be UAC, and the ribosome cannot read that. Any question about any of that? Viral genomes can be linear or circular. This is normal for eukaryote cells where they have linear genomes. And then prokaryotes have bacteria have circular genomes. And the virus can be either linear or circular depending on the species of virus. Some viruses have their genome separated into several segments. An example of that is the influenza virus, the virus that causes the flu. Their genome is separated into several segments. That should be normal to you because the human genome is separated into several segments, and we call each segment a chromosome. Any question about any of that? All right, when we're talking about the viral structure, it also includes the capsid, the protein coat of the viral, and if the virus has an envelope, it includes the viral envelope. As I stated, the capsid is made out of protein. It is composed of capsomeres. Let me blow this way up. For this virus, we only have one capsomere. And then that capsomere just repeats. I have no idea how many times, many times, to make the virus protein coat. Some viruses have several capsomeres. If the virus does not have an envelope, we call it a naked virus. So this virus here does not have an envelope, so it's a naked virus. If the virus has an envelope, we call it an enveloped virus. The envelope is a lipid layer. It does have mainly phospholipid in it, which forms the bilayer. And it does have proteins and carbohydrates in it as well. But the main molecule in the envelope is the phospholipid. Oops, didn't mean to do that. Let me go back. What I meant to do is shrink the size to get that all in there. If the virus has an envelope, it may be covered in glycoprotein spikes. So these reddish purple things that are protruding out of this virus are the spikes. Blow that up a little too far. The spikes are the outermost portion of this virus. The outermost portion of the virus is important because it's the outermost molecules of the virus that bind to the cell receptors. So all of the ligands and adhesions on this virus are on the spikes. The spikes are also important to the immune system of the host that gets infected by this virus, because the spikes are the furthest most out molecules of this virus. And the immune system will recognize the furthest molecules that are out on the virus and then mount an immune response against those molecules. So the molecules of the virus that the immune system will recognize will always be the outermost molecules. And on a spiked virus, those will be the molecules of the spike. Any question about any of that? Oh, I should mention that the molecules that the host immune system recognizes and then makes an immune response against, we call those molecules antigens. And it's the same molecules that serve for the attachment of this virus, meaning the molecules of the spike. For this virus, which has no spikes, where are the molecules which bind to its host cell? I'll give you a hint. It's the outermost molecules of the virus. Anyone gonna guess? Well, think about it. Where are the molecules of this virus which attach to the host cell receptor? Let's talk about the general morphology of the virus. It is part of the viral structure. The morphology is the shape of viruses. Viruses have three basic shapes, and they can be classified on the basis of their shape. The shape of a virus depends on the shape of the capsid. So you can have a helical virus, which has a helical capsid, and I can see it there. I don't really see the helix in the Ebola, so I don't know why they call that a helical virus, but it is. And then there can be a polyhedral virus, and this will be the shape of the capsid. It's sort of looking like a complex multi-sided dice. And there's an actual electron microscopic image of a polyhedral virus. This virus has spikes on it. And then the third general shape of a virus, we lump them all together and call them a complex virus. A complex virus can take on essentially any different shape. Here we're looking at a complex virus. There's another complex virus. And here's a complex virus. This one looks similar to the lunar lander from the Apollo space mission. And I know most of you weren't born when that happened, but you probably have seen videos of it or something about it, pictures of it. And there we actually see an electron microscopic image of this virus. Complex virus, taking on any shape. This virus is both complex and it contains an envelope. And there you can see the envelope, the reddish color going around that way. It's hard to see the envelope here, but it's right there. There you can see the nucleic acid inside this virus, and then there's the capsid right there. This, for some reason, this virus has two capsomeres, two capsids. This is a naked virus. It doesn't have an envelope around it, but there's the capsid. And we'll talk a little bit more about this virus. T4 virus is a member of the T-even family. T-even includes T2, T4, and T6. We'll talk a little bit about T4 because it is the virus which has been studied more than any other virus. More than any human virus, we have studied T4. T4 actually infects bacteria, and probably the reason why it was chosen to be the one studied the most is because it actually infects E. coli, a bacteria cell, which is very common in most microbiology labs. And T4 infects E. coli. Here you can see the virus. It has a head, which encodes the genome, which is a DNA molecule inside the head. And it has a tail, this lower portion of the virus. The tail helps the cell infect a bacteria cell. It does not carry the genome of the virus, but you can see that the head has a different capsid than the sheath of the tail or the inside portion of the tail. And that'll be a different protein than the pin and the base plate of the tail. And that will be a different protein than the tail fibers will be constructed of. So this has different capsomeres or different proteins that make up the capsid of this virus. Any question about any of that? All right, and that's an actual electron microscopic image of bacteriophage T4. That's the artist rendition. So what happens when a virus infects a host cell? The host will respond with their immune system. Infection of a virus with an animal cell stimulates the host immune system to produce antibodies. Antibodies recognize and bind to viral surface proteins that are always the furthest most out, why I've got the name surface in there. And these we call antigens, what the host recognizes as foreign and then mounts an immune response against. That is an antigen. Remember that the outermost molecules of a virus are also the molecules of the virus which bind to their host cell. So it's these molecules, adhesions or ligands, that bind to the host cell receptor. Some viral proteins can change or mutate frequently, where we get different viral strains that can infect the same individual more than once. An example of this is the flu. The flu mutates so rapidly and changes so rapidly that 3 years after getting your flu vaccine, that vaccine will no longer be effective. Now, in reality, let me show you a picture of that. I'm not seeing paint here. Is it there? Normally when I go there, it shows up paint right away, but I'll just do a search of it, go paint here. Oh, now I'm trying to remember, what am I trying to draw here? Normally when you're given an immune, meaning giving a vaccine, and this will be time over here on the x-axis. Move that out of the way. So this will be time. Oops, don't have it. Erase that. Time on the x-axis, and then your immunity, how strong your immune system is on the y-axis. So I'm just going to go percent immunity. The top being 100, the bottom being 0%. We can maybe do that here. Initially, when you're given a flu shot, your immunity will be quite high, but over time, that immunity will drop. Just trying to make a smooth curve there. That's not very smooth. And once you drop below a certain threshold level, they say that you have essentially no immunity and you need to get another flu shot. With the flu, your immunity drops from very high, if you're a young person, maybe 90%, down to a very low number within 3 years. And so your immunity does drop like this. It doesn't drop from here to there in one setting, or from here to 0 in one setting. It drops over time, and that time is in the order of years. So the first year after you have the flu shot, you still are protected, assuming the flu variant hasn't, a new variant hasn't come around. Another virus that changes fairly rapidly, and this is largely from mutation, is COVID-19. To have your immunity very high at COVID-19, you need to get the vaccine about every 6 months. But at some point, your immunity will drop below this level, in which case you'll have no protection from the vaccine. And I don't know when that is. It's probably a little over 3 years. They suggest with the COVID-19 that you get your vaccine about every year or sooner. Maybe some people are saying every 6 months, because COVID-19 does change and a new variant can arise, and the vaccine may not protect against that new vaccine. So far, all the variants that have arisen have been able to infect people, not all the variants that have arisen, all the late variants that have arisen have been able to affect someone who is vaccinated with the first vaccine. However, so far, the first vaccine has protected against severe disease, meaning you do get infected by COVID-19, but you do get a prevention of getting the COVID-19 developing to a very severe disease, which, if you remember, COVID-19 can kill people. Any question about any of that? All right, so the flu viruses changes very rapidly. We'll talk a little bit about why. And generally with the flu virus, you have to get the flu vaccine at least every 3 years. They actually recommend you get it every year. All right. Let's move on to the topic of taxonomy. Viruses are grouped into different groups, the same as other living organisms. However, viruses are only mainly grouped into families, genus, and viral species. You can group them into King Peter's son came over from. You can group viruses into orders, but higher groups, higher than order, are very hard to group them. And the reason is that viruses have a very small genome, and then their genome can evolve very quickly to adapt to the host cells that they are infecting. So viruses, because of their quick evolution and their very fast, their fast evolution and then their very small genome, means that we cannot classify them into higher orders most of the time beyond an order of the virus. One exception is we can group a virus into the kingdom of the cell that they infect, meaning we do group viruses into animal cells because they infect animal cells, and we do group them into bacterial viruses because they infect bacteria cells. This is not a kingdom of the virus, but it is a kingdom of the cell that the virus is infecting, and we can group them that way. Any question about any of that? With the viral species, we can also group them into subspecies. That would be like a human race, meaning the black race and the white race and the East Asian race. That's what a subspecies is. With the virus, though, the subspecies is very important for the virus. I think everybody knows about herpes simplex type 1 and herpes simplex type 2. Those are the same species of virus, but they are very different in the disease they cause. Herpes simplex type 1 causes cold sores, and herpes simplex type 2 causes more severe sores, and their genital sores. Herpes simplex virus type 2 tends to spread sexually by sexual reproduction. And then herpes simplex type 1 tends to spread from parents, especially the mother, to the offspring. But both viruses, strains of the virus, can spread both ways, meaning you can spread herpes simplex virus type 1 sexually, and it is possible to spread herpes simplex virus type 2 from mother to child, especially if the mother has a herpes simplex virus type 2 lesion on her genitals during childbirth. The child is likely to pick up herpes simplex virus type 2. Oftentimes, nowadays, if they see that being the case, they will do a cesarean section to prevent the child from being infected with herpes simplex virus type 2. Any question about any of that? let me talk a little bit more about how the subspecies of viruses are really important for the virus. In the poliovirus subspecies 1 causes the disease polio. And then the poliovirus subspecies 2 we use as the live vaccine to prevent patients from getting the disease polio. So one subspecies causes polio, the other is the vaccine that we give people to prevent them from getting the disease polio. Any question about any of that? That just shows you the importance of the viral subspecies. Oh, I need to say something else about the classification of viruses. Viruses do not have Latin names. So when you're naming a virus, you use common English names. Like the polio virus is the common English name for the virus, and the polio virus is used worldwide. Whatever language you speak, like in France, they call it the polio virus. What happened was, apparently the initial people studying viruses were all English speaking, and then they gave viruses common English names. And then the other languages of people who spoke different languages, who studied viruses, came in and they adopted those names. And so the common English names are all that are used for naming a virus. Any question about that? Do you guys like knowing that all you need to know for the virus is the common English name? You don't need to know a Latin name. Do you like that? Makes it easier. All right, so let's give an example of viral taxonomy. Viruses are separated into a family. One family is the herpes viridae, which is the name of the herpes virus family. So all family names end in viridae. The genus name for the herpes virus that I'm going to talk about is the simplex virus. So herpes simplex virus is the virus we're talking about. That's the genus. Then the common name will be herpes simplex, and then the subspecies would be herpes simplex virus type 1, or herpes simplex type 1. That is the common name. With the herpes simplex virus, scientists gave it a new name called human herpes virus. And this was done about 20 years ago. And I can tell you that the human herpes virus has not been adopted by most individuals. Most people, including scientists and doctors, continue to call it the human herpes simplex virus. So we're still using the old names. Any question about any of that? All right, so the viral family always ends in viridae. The genus ends in virus. The species will be a name like human herpes simplex virus. And then the subspecies is a strain of the virus, like human herpes simplex type 1. Here we're actually looking at a herpes virus. Any question about any of that? When we're growing bacteriophages in the lab, bacteriophages is the term used for a bacteriovirus. a virus that grows in bacteria cells. The viruses must be grown in bacteria. So what we do is, to grow up bacteriophages, is you make a plate of agar and you put the bacteria everywhere on the plate so the bacteria grow everywhere. We call that a lawn of bacteria. And let me blow this up. This off-white or white color is from the bacteria growing everywhere on the plate. The agar itself is the color of these plaques, a darker color. And then you, after you plate the bacteria, but before you incubate it, you then put on a solution containing the virus. And then you spread that solution over the plate. You then incubate the plate and note that the bacteria have grown everywhere except where the plaques are. Where the plaque is where a virus bound to the bacteria and then replicated in that bacteria cell. And what happened to the viruses that came out of that cell when it was lysed? They go to neighboring cells. And then they infect those cells and replicate those cells. And then they go burst that cell and go to out of that cell and they go to more neighboring cell. That gives rise to a plaque, which is seen here. And you can count how many plaque forming units were in the solution. If this was the direct solution that we made, then there would be, oh gosh, 100 to 200 viral plaque forming units per mil, assuming they put 1 mil of the solution on this plate. If you notice, this plaque and that plaque are larger than that plaque and that plaque. What may have happened was initially when the virus was plated on the plate, this virus here immediately touched a bacteria cell. And so this virus got started right away. Whereas these plaques, which are smaller, is that one there or is that something on my screen here? I think that's a plaque there. That plaque there and that plaque there and that plaque there are much smaller. What happened with these plaques is probably the virus went there, but there was no bacteria initially there. And what happened is the bacteria nearby grew, and eventually a bacteria grew to touch the plaque, the virus. And then the virus started replicating and then started forming the plaque. And that's why that plaque is smaller than this plaque or that plaque. This plaque is very large because it looks like it's a combination of 3 plaques that grew to form 1 plaque. Any questions about how you grow bacteriophages, viruses that infect bacteria, in the lab? I've actually done this where I grew viruses in the lab and formed plaques like that. So remember, bacteriophages need to grow in a living bacteria cell because that's their host cell. For viral multiplication, the viral genome only includes a few genes. And these genes are needed for the synthesis of the new virions. The virus replicates by hijacking the cell and then using the cell's components to replicate the virus. The cell's host cell machinery is used to replicate the virus. Like the ribosomes will replicate viral, will translate viral RNA to make the viral proteins. And then the virus doesn't make any ATP. It has no way to make ATP. It simply uses the ATP existing in the host cell. And the virus uses the host ribosomes, ATP, tRNA, amino acids, and some of the viral enzymes to replicate. The basic mechanism for viral replication is similar in all viruses. They replicate by infecting the host cell, hijacking the cell, and then using that cell to make new virions. In bacteria, bacteriophages can replicate in two life cycles, the lytic life cycle and the lysogenic life cycle. So bacteriophages only replicate in these two life cycles after they infect their host cell. In the lytic life cycle, the host cell is lysed and killed to release the newly replicated virions. In the lysogenic life cycle, the host cell remains alive for as long as the virus is in the lysogenic life cycle. Is that clear to everyone? Are there any questions about that?
So once the virus is killed, then the host cell dies in the second one?
We'll talk about the lysogenic life cycle next time. This lesson is going to end before I get to it. But the lysogenic life cycle, the host cell, which is infected with the virus, never is lysed and does not die for as long as the virus remains in the lysogenic life cycle. Okay. I'm wording it that way because the lysogenic virus can leave the lysogenic life cycle, in which case it will enter the lytic life cycle. And in the lytic life cycle, the virus will kill the host cell. Okay, but while the virus is staying in the lysogenic life cycle, it will not kill the host cell. And the virus and the host cell will live in symbiosis as long as the lysogenic life cycle continues. So the host cell is never killed or lysed in the lysogenic life cycle. All right, I think this is a good place to end the lesson. If I go further, I'm not going to be able to finish a topic before it's time to take a break. And oops. So I think I'm going to end it here. And before you leave, let me state that today there is no lab scheduled. In the lab today, you should be working on the infectious disease paper, which is due this Saturday. You should be working on your unknown project. I will be present in the lab for 15 minutes to answer questions. So I will be there from 7 o'clock to 7:15 to answer questions. But once the last question is answered and 7:15 arrives, I will log out. You do not need to come to the lab unless you have a question. You may come to the lab and just hear what any questions are, but I'm not going to guarantee anyone will come and have questions. But you can come to the lab to ask questions. Okay? But after 7:15, if there's no further questions, I'm going to log out. All right, any questions about any of that? All right, if not, I will see you in the lab if you show up. And if not, I will see you next Tuesday for the lecture. Bye.